for today, for our botanical seminar, we have our special guest, who is Gianluigi Ottaviani. I know that you can understand he's from Italy. He studied in Italy, but we met him first time in Australia where he worked as a research assistant. And now she came from Botanical Institute in Cheboň. So now he's settled in the Czech Republic. And his interests are in plant ecology, in functional diversity of plant communities, and today he will speak about biogeography of edaphic islands. Okay. So, Gianluigi, it's yours.
Thanks a lot. Thank you for inviting and kind words, and of course to all of you for attending. So yeah, today I would like to, can you hear me? Like voice? Okay, perfect. So I would like to present some key findings, let's say, and insights into...
Otherwise the students can move forward. Come.
But I don't know if it's hearable.
It's fine.
So yeah, I would like to present some key findings about in relation to a 3-year Gaccio project that is finishing, approaching the deadline in December, more or less, and which was funded by the Czech Science Foundation. And yeah, as the title is suggesting, I was focusing mainly on what may shape plant persistence strategies on edaphic islands, and I was using to try to implement the functional island biogeographic approach, which I hope I will be able to deliver, like the framework and some core results. So I would like to start with a brief overview of the talk. And so I will start with some basics into the island biogeography and also about plant persistence strategies, because as I think is evident also from the title, these are the 2 core ingredients of the recipe, and therefore how I got the idea to write the project. And then I would go into some more concrete results of the 3-year project, namely the conceptual framework. And then I will present some findings in relation to patterns of plant taxonomy, functional traits, and phylogeny in different edaphic island systems. And then I will delve more into intra and interspecific patterns of persistent traits within one specific edaphic island system, which is constituted by Trevic granite outcrops, which are located, I think, 50 kilometers from here. And then I would like to leave you with some take-home messages that I hope will be emerging naturally by the end of the lecture, and like insights of what I would like to do next. So this is a picture of some granite outcrops from Southwestern Australia, where I did. This is close to Esperance, which is, you can see the southern ocean in the background. So it's about 800 kilometers southeast of Perth, where I did my PhD. And these are, yeah, the outcrops. These are a bit outside of my study area, but I found this picture quite emblematic because it's showing the dome-shaped topography and how this, in fact, interbergs, which are terrestrial island-like systems, how they may look like. The ones in Trevic are a bit smaller, but you know they may operate as I hope I might be able to convince you similarly. And a few hints into the Southwestern Australia, let's say biogeography, ecology, evolutionary aspect is that it's one of the oldest and also nutrient poorest and most fire-prone region in the world. But it's also hosting one of the global biodiversity hotspots, because it contains more than 1,500 endemic species, and Southwestern Australia, the floristic region is pretty much in this part. And these are my study areas, and over there you can see the upper panel is about precipitation, and the lower is about temperature, and as you can see, there is a strong gradient running from the Mesic and cooler southwest towards arid and warmer northeast of the southwestern region. And then there, what I did, I was studying plant functional trait patterns along climate, and so I got the depth gradients. And in terms of climate, I was studying the relationship between a bunch of traits with aridity and with precipitation seasonality. So over there, this is a picture of 3 habitat types that I was studying on these outcrops. So herb fields that are growing on this extremely shallow soil, on pretty much on bare rocks. Then there are shrublands, and then Hebron woodlands. It's called Hebron woodlands because these are growing right at the edge between the outcrops with the surrounding landscapes, and they are receiving this extra nutrient from the runoff on the almost impermeable slopes. And they are quite different also from the ecological point of view because they are, the soil rejuvenation process is quite active and happening compared to the rest, which is quite nutrient poor and stable. So there I was studying some above-ground functional traits, which are definitely related to two of the major drivers in the region, the putative major drivers in the region, which are resource status and fire regimes. And these are, in fact, related to leaves, to architecture and bark features. And then, after this is brief brackets into my past, and more remote past, and now it's more recent past, I moved to, after my PhD, I got postdoc position with Itka Timeshova at the Institute of Botany of the Czech Academy of Sciences in Trebon, where I'm still working. And yeah, these are pictures from Trebon in South Bohemia, where I moved. And what I was doing there, I was focusing mainly on a group of different groups of traits which are largely affecting individual plant persistence strategies, and which are mainly generally played below ground, but not only, but mainly below ground. And here there are some examples. For example, we've got a bunch of longevity traits, for example, the age of a plant, which I will show more information later on, or radial growth. They can inform about how plants can for how long an individual plant can persist in a given spot. And then there is like the space occupancy of an individual, let's say, functional individual or a clone, which can be formed by variety, by number of interconnected gametes. How they can spread and multiply in a space can also affect the local persistence. And then, of course, whether or not and how much the plant is able to recover after the damage, which can be generated by climate adversities, drought or cold, but also by disturbance, mowing, fire, whatever, and which is tightly related to the availability and protection of bud banks, and also on the availability of carbohydrate storage and different types of carbo storage. So here it's another picture which may exemplify whether having or not having clonal ability can affect competitive ability, multiplication, nutrient acquisition, storage, and resprouting in different species. This is a picture from Itka's paper. So I think it's necessary. I would like to, I will try to avoid jargon as much as possible. Hopefully I will succeed, I'm not sure, but this is unavoidable. So we must be on the same page. Well, we can disagree, of course, but I need to explain to you what is a functional trait, and now I'm using functional traits. Because otherwise everything, we are missing one key pillar. So what is a functional trait? So tradition has been defined as any morphological, anatomical, phenological, and potential behavioral feature which is informing on specific functions exerted by plants, and we would add also on challenges that are faced by plants that may largely affect their performance and fitness, and these are typically collected or measured at the individual level. And plants, definitely being sessile organisms, they are performing a variety of different functions simultaneously. And this includes acquisition of resources, conservation, use, and of course in clonal plants also sharing of these resources. If they have bud banks, the ability to resprout after disturbance, and again if they are clonal, the ability to explore and occupy an area, and then finally to reproduce either sexually or vegetatively or both, and for the end to disperse propagates. And because plants are inhabiting finite resources environment, you cannot have it all at the same time. So traits are generally co-varying positively, or they may trade off, so varying negatively, having like functional spectrum, or sometimes they can also vary independently, so identifying separate functional axes. And this is like an example from a paper in Ecology Letters, where here there are a bunch of acronyms, but for simplicity, here you can see that the red arrows identify the STEM economic spectrum on one axis, and then there is the green axis, is about leaf economic spectrum, which is orthogonal to the other one, to the STEM economic spectrum, and then there is a third one, which is the leaf area spectrum. And this is run, I think, on more than 700 species from tropical regions. Okay, so how did I get to the basic idea of the Gatcher, of the junior researcher Gatcher's proposals? So I wanted to somehow link what I was doing during my PhD, working with Inselberg, so terrestrial island-like systems, with persistent strategies. And therefore I was thinking, okay, why not examining what may promote or eventually hinder local persistence of plants on islands or insular systems? And why? Most importantly, because of course, if it's just my curiosity, who cares? But why should it go that way? So first, why islands? Well, islands have been definitely a research focus for centuries, and starting even before than Wallace and Darwin, but definitely they have been considered, they're still considered, and they constitute excellent models to examine potential putative ecoevolutionary processes which are shaping the dynamics and formation of communities or entire flora or biota on an island, because these ones are considered simpler systems than mainlands because they're being actually spatially confined. And also, their islands generally are very rich in endemics, and the biota that can be observed on islands are generally quite, it's called disharmonic or distinct, or they're having different features compared to the mainland counterparts, and this can be captured eventually through trade values, combination diversity that I will be talking later on. And this distinct feature, so like island distinctiveness is also called term island syndrome, following Carquist's 1974 book. And traditionally, the classic research on islands has been focusing on patterns of species richness and diversity, and how these are changing actually with island area and with distance from mainland. And this is spatial isolation. So this is the classic book from MacArthur and Wilson, where this island size and spatial isolation are the core drivers. And this is one example of this relationship, like the species area relationship, which was in fact, I think, first observed or documented on islands, and which is showing how bigger islands can host higher number of species, because larger areas can host larger populations, which may eventually offset extinction risks, but also larger islands can potentially host higher diversity of habitats, and this can in turn affect species diversity because there is positive correlation between the two. And traditionally, in fact, the focus of the research on island biogeography is on three main processes, and these are colonization, extinction, and speciation. And this is like a nice graph from Warren et al., showing how the role of ecology is declining over time, whereas the role of evolution is increasing over time. And here the authors are representing, showing putative measure processes which are interacting or like interplaying or shifting over time, and how this relates to the number of species. As a result, persistence is overlooked in terms that is not directly addressed, because it's somewhere there, because, for example, persistence can be also seen. If a species is there, it means that it's not going extinct, of course. So, or like the island syndrome can also inform about the potential persistent strategies that species may put into place to stick around, but is not really in the picture, as this nice graph from Whittaker et al. Is showing. So we may say that definitely persistence is somewhat overlooked on islands. And now we are entering a bit more into the details of the project. So here I put two papers that appeared recently. One is from our team, another one is from Julian Schrader and colleagues, where we are trying to condense ideas into this field of what we have been calling functional and biogeography, which means simply what can we gain by implementing trait-based approaches, so studying how traits are varying, different traits are varying in each system on islands, let's say. And a good example is represented by the island rule. So the island rule, for simplicity, is like the tendency of small organisms to become giant on islands, and the opposite happening for bigger organisms tending to become smaller, so the dwarfism on islands. So this is like I put here like an example from a recent work by Vidic and colleagues, showing how woody plants in the upper panel and forbs on the lower panels are actually aligning, obeying, how they are saying, or disobeying to the island rule. As you can see, for the plant height, there is pretty much no, they are nicely obeying the island rule. Whereas for leaf size, we've got woody plants which are pretty much obeying, whereas forbs are not obeying, and seed size is actually not following the expected patterns at all. And that's where we believe it's very interesting to study why they are not following this pattern. So here I don't, of course, want you to go through the entire list of trait functions, but definitely this is to say that, okay, by using traits, as I was saying before, we are interested on the function that you can capture by studying traits, so which specific challenge you can proxy by measuring traits. So here we are saying, okay, we've worked through a list of functions, and therefore, and then I'll show you a list of traits which we believe is important to consider while working on islands, but of course could be also implemented on non-mainland systems. So from dispersal ability to hotspot persistence and so on and so forth. And then we here reported the list of traits, potential traits that one can pick up either to go and collect in the field, or if data are available from existing databases. So it doesn't mean that one should collect the whole thing, or depending on the research question, one can, for example, be interested only on dispersal ability, so one can work with one or two dispersal traits, dispersal ability trait, or if one is more interested on the ability of plants to occupy space, can focus more on these other traits associated with space occupancy. So, but if one is interested on the entire syndrome, functional syndrome of the plants on the insular system, well, that's more tricky. We should collect at least one trait for each. We should tick every single box with one trait per function at least. So as I was saying, like by using trait-based approaches, we can tackle, or we can address some quite a lot actually, quite a number, more than 30. According to Schrader, they identified 33 hypotheses and questions that can be tackled. Patino, they identified 50 questions which are most pressing since 50 years since the formulation of the theory of island biogeography. And we have identified that about 35 out of those can be tackled by implementing trait-based approaches. So by using by analyzing traits, we can try and give some questions relevant for island biogeography. And here I would like to bring in some examples. This is from our paper, and we are saying we instead identified only 3 major hypotheses. We are like a bit more reduced. So we are saying that insular systems are characterized by unique plant functional syndrome. So this is like this idea of island syndrome. And here we are showing like what we are expecting in a simplified way, of course, the difference between mainland plants and their island counterparts. So we should be observing on islands that the plants are more limited in their dispersal, producing larger and fewer seeds. They may be losing spinescence, and then this island rule that I was explaining before. Another one could be how plant functional syndromes are varying along insularity gradients, namely if the variation is predictable. And imagining that we should be islands which are, for example, bigger and closer to the mainland should be showing less discrepant, less distinct features compared to islands which are most farther and more and smaller compared to the other one. So that the distinctiveness of being on island should be bigger with increasing insularity. And now we are going into some results associated with really related with our data from the project. So the first one is about taxonomic aspect. So we studied edaphic island specialist richness versus insularity, and we focus as model system on 3 edaphic islands systems. One are the spring Calcareous fens in Western Carpathians. Another ones are the Trebich Outcrops here in the Czech Republic, and the third one being the mountain tops in Cantabria, Spain. And the basic question that we wanted to answer was like, if we can assume that edaphic islands are ruled by similar biogeographic forces as true, let's say, oceanic islands. And these are definitely, like, once we entered that field, we realized that, okay, first of all, what is mainland? What does it mean mainland for terrestrial island-like systems? So the first challenge to overcome was to identify putative species sources in a system, and borrowing concept and notion from island biogeography associated with mainland, we identified those patches which were the largest and also richest in specialists as putative species sources. And now I'm not going into the methodological details, but this has been corrected also according to topological mismatches, and so I'm not going into that. But if you would like to have some questions, if you want to have questions, I will be happy. Hopefully I will manage to answer. And by largest and specialist richest, I mean we have used the data distribution of island size or edaphic island size and specialist richness, taking the last quartile, like about 75%. And then from there, we could calculate a comprehensive set of insularity metrics. Some of those are reported in this image from Weigelt and Kraft. And you cannot read it, but I wouldn't want you to read. But the thing is that there are a bunch of potential metrics that are related to insularity, but the major groups are associated with island network, so the connectivity of the islands, the distance from the species source, and island size. So then we calculated 9 or 10 metrics, and again, I'm not going into all the stats methods, but then if you have questions, please go ahead. And so we identified which are the best predictors of specialist richness for each of the system. And as you can see, in fence, we identified island size and target effect as the most effective, the most parsimonious as well, the most parsimonious models. And then for mountain tops, we identified only size as best predictor of specialist richness, and in outcrops, target effect, even though it's not super strong as an effect. So this, let's say, island size, target effect, and isolation can be considered as the best predictors for specialist richness in these study systems. And target effect, as you have seen here, it's a simple quotient between isolation and size. So here, I would like to spend a minute or two explaining what is target effect. So this is a picture drawing that Francisco, during postdoc project, elaborated during the production of this paper, tried to exemplify and visualize how it works. So target effect is, as I say, is a combined effect of spatial isolation, so distance from the species source, and the size of the target island. So, of course, closer, let's say, being closer, it enhances the possibility of an island to be colonized, but then if the island is smaller, of course, the chances are lower. And this is interplaying once you are starting to move around with larger islands, more smaller island, and with distance. So as you can see here, then like the farthermost and the smallest target is becoming almost unreachable or hardly reachable. So it's pretty much target effect telling us how hard or how easy it is to reach and to hit an island by chance from dispersing propagates. Also, we found in one of our paper that actually target effect is also able to capture the multidimensionality nature of insularity, namely that it is not only related to island size and isolation, but also to these other metrics that I was mentioning before, especially the connectivity and connectivity metrics. So here I reported the result from a PCA that was run, so a multivariate analysis, reporting, don't bother about the acronyms, but these are all a bunch of insularity metrics, 9, that have been measured for that system, for Granite outcrops, but of course excluding target effect. And then what we have done is that we took the PCA score of the first axis, which is explaining quite a lot of the variability, and regressed with target effect. And as you can see, the relationship is quite strong and tight. Okay, so now we move from more taxonomic cut into more functional aspect, and presenting some results about trait average and diversity versus insularity. And here is the map as before. Well, it's been modified, and still the three systems from fens, outcrops, and mountaintops. And the overarching question was whether local persistent stability of edaphic island plants are actually positively correlated with insularity. And again, here I report list of traits and functions that we have been using by extracting them from available databases such as Pladias, Global, LEDA, and others, associated with persistent strategies. So, as you can see, there is a Rankier life form which is informing on the multifunctional persistent strategy, but it's quite, it's categorical, so it's good trait, but definitely it's a bit rough. And then there are reproductive types, seed mass, size of the below ground bud bank, depth of the below ground bud bank, and so on and so forth. So these are informing on the variety of functions which can largely affect the ability of plants to persist or not on a spot. So some results. So here we have on the y-axis the trait means, and here I will be presenting some results that are selected just to because there are way more, but I think in order to make it a bit easier, like the story flow, I think it's good to focus on some of those that we consider more relevant. So here we are showing like how trait means, for example, of lateral spread, which is how far is able like an offspring ramette. What is the distance from the parent plant and the offspring ramette? So it's telling us that it's a proxy of the clonal ability and how much plant is able to occupy a space. So in this case, as we can see, lateral spread is declining with island size, whereas is increasing with isolation and target effect. Target effect being this comprehensive insular metrics. So this is quite well aligning with our expectation, because it means that plants on smaller islands, they are less able to spread, but the opposite is true for like highlands, for sites which are more insular and isolated. And when we go for functional diversity, we found, okay, here we have different traits. Here we got the diversity of type of coronal growth organs. So type of coronal growth organs means in coronal plants there are a variety of different organs which may contribute or exert the vegetative reproduction, namely rhizomes, which are stem-derived, then there are stolons, again stem-derived but above ground, whereas rhizomes are below ground. Then there are root sproutings, so roots which are having buds and potentially can become independent ramette. And then there are bulbs, root splitters, so there are a variety of coronal growth organs. And here we measure the diversity of these coronal growth organs, whereas the last one is the diversity of branchial life form. And here actually we are observing the opposite patterns as before. So the diversity of, for example, coronal growth organs is increasing with island size, and whereas is decreasing with isolation. And the same happens for the diversity of branchial life form with Tiger effect, meaning that if we combine these results with the other ones of the mean values, well, we can summarize saying that. So back to the answer to the overarching question about our local persistence abilities of edaphic island plant specialists positively correlated with insularity, we can say yes, because with stronger insularity, persistent related traits tend to get higher values, for example, spreading more laterally, more and having more and better protected buds. I haven't shown this, but they are following this pattern, but they're showing less diverse values. So this it means that they have better abilities to persist locally through similar and possibly fine-tuned, meaning possibly adaptive values. And now we move into a bit more evolutionary cut. So we would like, these are like new, the other results I think have been seen also by the authors, here some of them. And this one instead are quite, it's the first time that put them together because they're fresh. So it's a work in progress led by Adam Klimesh, his postdoc with the project. And we want to know if there is if phylogeny is playing a role in shaping edaphic island specialist assemblages. And yes for fans, no for outcrops. So here, like there is the phylogenetic tree, showing that the fan specialists are very much clustered towards a bunch of families, most importantly Cyperaceae and Juncaceae. Whereas for outcrop specialists, it's pretty much all over the place. And this is, of course, confirmed by the test. Again, here I'm not presenting that much into anything into the methodology, but I'm happy to answer eventually. So, as I said, yes, are they structured? Yes, they are structured for fans, but nothing happening for outcrops. And then we want to know also whether and how the phylogenetic diversity of the edaphic island specialist is related to insularity. And this is quite interesting because it's declining for the phylogenetic diversity, is declining for fans, whereas it's strongly positive for outcrops. And for fans, interestingly, we have got here, like I put this dashed line, it means like significance at alpha 0.5. So all these blue dots here are actually island sites that are significantly phylogenetically clustered. So this is kind of confirming what we have seen also from the phylogenetic tree, and also partly aligning with what we are seeing from the functional analysis before. So there is a constant clustering, functional and phylogenetic with increasing insularity. And then we wanted to inspect for two traits associated with abilities and therefore to persistence, whether phylogeny is affecting the relationship between percentage of coronal species site level, and for lateral spreading that I will present after this. And as you can see, the outcrops are pretty much not affected by phylogeny, and this is again, I think, well corroborated also from the phylogenetic tree. Whereas, for fans, we are observing that these are different approaches of considering phylogenetic effect, but for the full randomization, it's a full Brownian motion, actually, we are observing the pattern disappear, the positive pattern that we can observe, that, for example, with increasing insularity, we tend to observe an increase of percentage of Granite species, but this is, once we correct for phylogeny, this pattern disappear for the full, for the full model, let's say, for the full randomization. Whereas, if it's an intermediate approach, pretty much is aligning with the non-corrected pattern. And it's also interesting that, okay, this is, if I remember correctly, this is not significant pattern, whereas that one is marginally or significant. Instead, for lateral spread, we have both positive relationship between, of course, with insularity. So, like as we have seen before, a stronger insularity implies that plants are, there is a positive correlation. So we cannot say that because of that, but definitely there is a tendency of better coronal abilities through lateral spread in more insular sites. But once we fully correct for phylogeny in fans, we actually observe the opposite. So we say, okay, so what? Let's have a look. In fact, we say for fans it seems so, but it's in X, whereas there is no effect for outcrops. So this is the outcome. Once, because Adam, while he was running the analysis, he realized that there was this equisetum variegatum that is very, that actually is doing a lot in the least insular part, and if we remove that, actually the effect of phylogeny disappears. So, of course, I'm not saying that we should go just focusing on this, but I think it's quite interesting to see how the weight that this single species can have on the entire pattern. It can really change the world. Okay, so now heading towards the last bit of related to the project. So here now I would like to delve into the Trebitsch system. So working at the intra and interspecific level of a bunch of persistent traits. And here again, the overarching question is, are edaphic island plant specialists, in this case coronal and non-coronal perennial herbs, distinguished by enhanced persistence abilities under stronger insularity and harsher soil conditions? And here the panel B and C are showing the outcrops. So they are a bit different from the ones I was showing in the first image from Southwestern Australia, but still they are insular or partially insular. And so here, like, it's more like the B panel is more like the landscape, how it looks like in the landscape, and the C is the habitat type, so this shallow soil acidophilous grasslands. And here there are two examples of one coronal species, Caret caryophylla and Carina cauris. And I would like to stress that here we work with 13 species, 5 coronal, 8 non-coronals, which corresponds, even though the number doesn't look so great, so high, but they correspond to about 45% of the entire specialist richness of the edaphic island system. Again, the traits and the function, so traits that we have been collecting and measuring within and across species, and the different functions which are related to persistence are here reported. So, and as you can see, age, radial growth, storage tissue, vessel size, these ones are quite rarely used in functional ecology in general, and I know of no study at all of examining these traits for on island systems. And generally age, for example, is used because these ones are coming from the growth rings of the herbs, because perennial plants can make, herbs can, not all of them, but most of them are able to, especially dicots, are able to make growth rings, and then I will show you some picture. And from there, from these slide sections, cross-section, we were able to quantify a variety of anatomical traits, and from them we have selected the ones that we believe are more informative. Of course, some of those were plant height and below ground organ matter content have been collected for both the groups, clonal and clonals, whereas lateral spread is exclusive to clonals. And the anatomical traits from age, radial growth, storage, tissue, and vessel size are only for non-clonal plants, because collecting the oldest part of the clonal plant, well, good luck. Because, just to explain this, because either it's decomposed or you may need to dig, I don't know, 1 hectare of, so it can be very, very tricky. So it's doable. For some species it's possible. For the vast majority of species it's really, really tough. Okay, this is like, these are some results. In the upper panel, there are clonal species, in the lower panel, non-clonal species. So this is like how the different species are plotting in a multivariate space created by the traits. So as you can see, the clonal species tend to be more consistent in their occupancy, even though there is coileria macrantha, so which is characterized by consistently higher plants. The others tend to be nicely lumped. Whereas for non-clonal species, it's very evident already here that they are pretty much occupying different niches in the multivariate space. And this will be better presented here, because now we'll present some results about bivariate relationships. In this case, I present how insularity, the relationship between insularity and below ground resource conservation captured by this below ground matter content, below ground organ matter content. And here are the 5 species, 5 clonal species, sorry, which are consistently and positively associated with insularity. And the relationship is really, really strong. Meaning that, of course, the clonal species tend to be more conservative in their resource below ground, resource use and conservation in more insular conditions. Another example, this one is not in relation to insularity, but in relation to soil depth variability, which can be seen as a proxy of macrohabitat conditions, which again, all the 5 species, all the 5 clonal species, are behaving in a very similar way, so that they are becoming more and more conservative towards more variable and heterogeneous soil depth. But for these are, I don't want you to go through the table, but just to show you, these are for non-clonal species, 8 non-clonal species, and from already here, it's easy to realize that there are some species, for example, Carlina, Edianthemum, and Lignis, which are very much, they are responding quite a lot, whereas others like Nautsia and Silene and Thymus, which are almost insensitive, unresponsive. Not only that, from the distribution of the relationship here, you can see that there are some species which are tending to respond more to soil parameters, whereas others are more responding to climate, pretty much nothing, or very little, but also there are some which are very nicely responding to insularity. So we can say that non-clonal species, their responses are very highly species specific to variation in soil, climate, and insularity, with some being very, very, very responsive, while others are almost completely unresponsive. So to wrap up and back to the basic question, so if edaphic islands are distinguished by enhanced persistence abilities under stronger insularity and harsher soil conditions, yes, we can say. And clonal species are more consistent in their responses, both intra and interspecifically, and they're showing more resource conservative strategies with stronger insularity and harsher soil conditions. And this is the result here. This is again just to exemplify pretty much what I was showing in the previous multivariate analysis. This is hypervolumes, but here you can see that coileria is the green one, is the only one which is doing a bit different things in relation to plant height, whereas the others are all lambda. Instead, for non-clonal species, these ones are exhibiting distinct and species-specific responses to variation in solarity and environmental conditions. And this is, again, the hypervolume analysis that, again, it's just for visualization in this case, but you can see that these species are really doing in this trade-by-trade relationship in the United spaces. Really, they are really nicely segregating. And the last chapter, paper of the work in progress paper in relation to the project is about plant growth and age relationship with climate fluctuation. So we wanted to know, we were interested to see if actually selected perennial non-clonal herb species which are specialized to these dry grasslands offered by the edaphic islands which are operating as biological data loggers, meaning if they're able to capture the climate fluctuations over years. And these are the 4 species that we have been selecting. We have been selecting them not because we love them particularly, but because these guys are able to, they are offering a reliable chronology. So this is like a technique which is called herb chronology, which is borrowed, was originally developed for woody species, then the chronology, which has been then implemented also to herbs, for these ones that are able to create growth rings. So there are techniques, it's completely not my cup of tea, but our colleagues, Hans Hartmann and Diri Dolja, they are skilled with this stuff, and they made reliable chronology for Helianthemum grandiforum, Silene nutans, Thymus pullegioides, and Lichnus viscaria. So these are how they look like above ground, and this is like the examples, some examples of this cross section with the yellow lines highlighting years, or yearly growth. Unfortunately, we had from the 8 possible non-clonal species, we could have reliable chronology only for these 4. Knautzia was borderline, but we didn't trust it. So here are some results. So there are some, this is about relationship between establishment over the last 20-30 years and exactly, and which are the most important factor, monthly climate factor. These are the blue is precipitation, the red is temperature, and the yellow is the SPEI, which is an aridity index. And here you can see that this one, Helianthemum and Silene, are deep-rooted heavy-seeded species, whereas the Lichnus and Thymus are shallow-rooted light species seeded. And as you can see, the 2 upper panels, they are quite responsive, especially to winter and summer drought and temperature and precipitation. Sorry. Whereas the other 2, the establishment is almost completely unrelated. And these ones on the right panels actually are showing some, which we are still amazed by this relationship, because this is like the black line actually is the plant response, and the yellow, the colored lines instead are the selected climate parameters, and it's crazy. They are very much going raised together. And this is instead for growth. Again, the deep-rooted, heavy-seeded species are doing quite different things compared to shallow-rooted, light-seeded species. And to wrap up here, I think it's better so to put the whole things together from these 2 quite difficult graphs. So we can say that Helianthemum and Silene, which are deep-rooted, well, their establishment, deep-rooted and heavy-seeded, their establishment was positively affected by winter and early spring precipitation and aridity, while their growth was more affected by the winter conditions. Whereas for Lichnus viscaria and Thymus pullegioides, which are shallow-rooted and light-seeded species, their establishment was weakly, if anything, if at all, influenced by climate fluctuation, but their growth was actually very much adverse by high summer temperatures. So even though they are all perennial, non-clonal, inhabiting the same habitat type, they are responding very, very different. And actually, Lychnis, Viscaria, and Silene nutans are even belonging to the same family, right? Silene and Lychnis, I think it's the same family. So actually, we can say that in order to answer the question, we can say that yes, they are able, so these perennial non-clonal species specialized to these conditions can serve as biological data loggers, but in two different ways, according to their rooting depth, light seed, and seed mass. So now I'm heading towards the take-home messages. The first slide is a bit more general, and the second one will be a bit more into the results. So one is about the terrestrial island-like systems, such as the studied edaphic islands, can be governed or may be governed by similar forces as those which are operating on true, such as oceanic islands. And insularity is not a binary feature, in essence. It's not like either is insular or non-insular, but can form gradient. And these gradients can operate both within as well as across systems. And that target effect emerged as a comprehensive, I would say relatively easy to measure metric. It's not an easy to measure absolutely, but in comparison with other metrics, so the metrics such as stepping stone path or area, available area. I mean, there are a bunch of metrics which are very, very much time consuming, whereas target effect is basically, as I was saying before, is basically generated by island size and isolation. And therefore, this target effect it's able to capture multidimensionality or insularity as well. And that studying persistent related traits in insular system can help to understand how different species and also life histories may respond to variation in insularity as well as other environmental factors. And then that our findings suggest that plants specialized to edaphic islands can play out a variety of fine-tuned strategies that may boost their local persistence with stronger insularity. And this can happen at various scales, from the individual to population to species and to site. Phylogeny and therefore evolutionary history seem to play a role in shaping insular specialization and may also eventually affect trait patterns versus insularity, especially for ferns, but not much so for outcrops. And we will see what happens once we will have results from mountaintops, which are on the way. And that persistent strategies can largely differ among life histories. The highly consistent responses of clonal plants to insularity and soil conditions may indicate that these species are possibly more prone to local extinction risk, whether, for example, they occur like changes in the landscape configuration or land use may happen. Yet they are also quite, it's well known that clonal species are able to persist for very long periods even. So the extinction depth can be quite pronounced. And the end, that highly diversified and species-specific responses of non-clonal plants suggest instead that while some species may go extinct, others may be able to stick around, and this is also supported by, seems to be supported by results from the growth ring analysis that was the last study. And that being said, I would like to conclude with what could be next. What I would like to do next is I would like to stick to edaphic islands, but in this case, to the ultimate, let's say, to the prime system, which would be constituted by serpentine serpentines, on the quest for the syndrome, the island syndrome on serpentines. And this I would like to eventually work on mainland as well as on oceanic island worldwide. So this map, I think, it's quite cool because it's showing where actually serpentines are occurring. So these serpentines are this ultramafic soil. Well, serpentine is a type of rock which geologists are not very happy with because it's very broad. So anyway, from ecological point of view, I think it's good enough. So serpentines are ultramafic rocks which are outcropping in various parts of the world, as well as they are deep in the oceans, and they are generated generally by subduction zones, or where there are contacts between different tectonic plates. And from an ecological point of view, serpentine soils are actually generate a very toxic and highly selective environment for the biota, for plants inhabiting on top. And they offer very nice setting for island biographic studies. And I've got already an ongoing collaboration with these guys, the first being Fernando Silveira from Belo Horizonte in Minas Gerais, and they will be happy to work on those, what they call cangas, so these ultramafic outcrops in Brazil. Another one being Tomai Banet from New Caledonia. He's also a global biodiversity hotspot, and especially in the main, I don't remember the name of the main island, in New Caledonia, there is a nice separation between outcrops of serpentines and the rest. Another one in California, Susan Harrison, that she has been working on serpentines with her colleagues for, I think, more than 30 years. And she's got a lot of knowledge and data to share. And maybe this one you can recognize. Is Giamma from that may help with the Italian part, because there are very interesting systems in Tuscany and its archipelago, because so there are serpentines which are outcrops which are widespread or distributed, partially distributed in the mainland as well as in the archipelago, Tuscan archipelago, actually till ports. And with this I conclude. I would like to conclude, and definitely I'm thankful to all the people, the members of the team, especially Francisco, Luisa, Adam, and the technicians, Vercha and Hanka, for helping with the analysis or with writing, discussing, and of course technicians more for the lab and field work, and of course to all the amazing research network and group from Mitka, Milan, Mikael, and David, Borka, all of those. And of course, Czech Science Foundation for supporting me, and as well as Institute of Botany for the logistic, infrastructure, and money. And you all for attending. And yes, last, that's some pictures from these anatomical cross sections. So this is how Thymus perugeus looks like. This is Cleranthus. The black, just for the black image is like the same one as here, with using polarized lens that sometimes is useful for deciphering growth rings. Like in this case, for this Cnis biscaria, it's much easier to see the growth rings in the polarized lens. This is Nutans, Centaura stebe, Helianthemum. That's it. Thank you.
Thank you, Gianluigi. You have really a bunch of results. I think there will
be hopefully I wasn't killing.
Yeah, nice results. So, your comments or questions? So I will start with the first one. So you studied several traits. You showed us a table of traits. There are several ones, but you focused on those which are related to the persistence of plants. But I would be curious how the species, how the traits which are related to dispersal of the species,
yes,
will correspond to some insular habitats.
Well, one about dispersal is included in Luisa's paper, because we have seed mass, which is, of course, is not the best dispersal trait, but I agree. I mean, this is, and especially if there is a trade-off between persistence and dispersal. That's definitely something that could be inspected and will be inspected. But for such stories, we tried really to stick, because dispersal, as I was saying before, is colonization process through dispersal is one of those processes which has been really well studied, but definitely how it may relate, how it may trade off potentially with persistence is definitely something that is very interesting to look at. I agree.
Yeah.
Thanks for a nice talk. And I have a question related to the phylogeny correction, because I'm not sure if this is a necessary thing to do, like for the real islands, when the biota evolve on the island, so if you have just one ancestor, so all the descendants are just related phylogenetically, so it replicates the pattern. But here on these island-like systems, they evolve somewhere else. So I can see, or I would score every single species like independent process, independent colonization. So having 5 related species, it's probably, I wouldn't, corrected it as a pseudo-replication, scored it as a pseudo-replication, because they are definitely independent evolution or colonization events. So what do you think?
Well, so you mean that if we correct by phylogeny, we may fall into pseudo-replication issue, you mean?
No, I mean that doing the correction doesn't make sense to me, because you are correcting for something which is not happening on those systems. So you are doing the corrections because you're assuming that phylogenetically related species are not independent, are not having the same value, not our independent observation. But I think this is true for real island. When you have, then when the biota, most of the biota evolve in that island. So if there's just one ancestor, so all descendants.
You mean because then they
are
radiate?
Replications? Yeah, and just because the evolution happened, so you have, so in one island you can see a correlation between some trait and the island characteristics, and it might be just a matter of chance that this ancestor was lucky enough to evolve more descendants. But here they evolve somewhere else, so having even closely related species on one island, they had to colonize the island independently. So for me, these are
I think
independent.
it makes sense to ask if there are traits which are in several lineages which enable those species to colonize this new habitat.
Yeah.
So is it a question if there is some trait which is somehow conserved in one part of the tree, or if this adaptation is possible from several. I think this is such a question makes sense.
So,
well, it depends. Why are you doing the phylogenetic correction?
Okay, the story here of this paper is still a bit work in progress, because as you see,
that's why I'm asking.
Exactly,
and
the question is really that we are curious to know if there is a role, especially in the shaping of this persistent patterns, if there is a role for phylogeny in the expression of this trait, which are partly related to persistence. Namely, that the pattern that we have observed in Luisa's paper, in the other paper, it's really, let's say, something that we can reliably associate and say, okay, promote this or don't promote because of insularity, or because there is also something like an evolutionary effect that can help explain. It looks like, in fact, there is not, or very little, because also for the other one, which was lateral spread, once we exclude one species, which is doing all the thing, because it's, and actually Adam is running this 3D, so this is non-corrected, this is kind of mid-ground, midpoint, and this is the full correction with Brownian motion, and this is the one which is really messing up the thing. So when it's the midpoint, kind of, it's this is really technical, the technicality that Adam did, we can really ask him better. But definitely what he's showing is that really there is not big effect of phylogeny. So that what we have, the pattern that we have observed in Luisa's paper, it's corroborated also from this phylogenic approach. Another thing is about this other image, the regional assemblage of the island assemblage. This one, I think this is quite interesting, even though, of course, it's quite,
this is absolutely interesting, to see that the species inhabiting one island systems are phylogenetically related. So it's a set of traits that define them to be ready to utilize that environment.
Because probably this is very much related not to the strong edaphic filter, or let's say. So these are because they're simply the best in those systems.
But okay, but here's my concern with that. with that analysis is that you're not controlling for the very different habitat types. So you're getting all these cyperaceae. Well, no kidding, because you're dealing with open, wet grasslands. So it's not an issue of insularity. It's an issue of open, wet grasslands. So the problem is, how do you know that this is insularity versus...
No, this is not insularity. We are not saying insularity. So here we are saying this is the structuring of the edaphic kind of specialist assemblies.
But you're interpreting this through the lens of insularity. And I'm saying, I don't think insularity has anything to do with the differences you're seeing here.
No, I agree, and probably it was I didn't explain properly. Okay. So this is something else. I agree that this is about, these blue ones are the fence, especially the green one. So we are showing simply here that the assemblages of the edaphic kind of specialists are clustered in fence and not clustered or like random actually in outcrops. Then we went to inspect the potential role of insularity with the next analysis, and we may make some inference, maybe justify it, maybe not justify it, according to, for example, in this clustering, what I was saying before, maybe that was the confused, this one. When I was saying that these are phylogenetically clustered, significantly clustered, all this fence here, there are a few tens.
Right.
And I was saying, probably that's where I confused that this might be also associated with the structuring in the assemblage. But right. Maybe also not the case.
So I would think that if you're going to start looking at insularity, what you would want to do is compare against a null expectation for a non-insular habitat and show that there's a difference. So if you were to take these fins and compare the patterns to, say, some kind of riparian system that is connected and show a difference,
well,
then we can talk insularity.
I agree. This one, production as we did for Luisa, these are for specialists only. So they are the matrix, so we can see what happens with the other ones. But you know, I think.
Well, but
again, these are very preliminary.
If you were to look at specialists of riparian wetlands, what would that look like?
Well, that's on it's if.
I mean, but you'd need to show that riparian systems that are connected have a different pattern than the insular ones for you to actually invoke insularity.
But we mean with this one or with the other one?
With this one. Without that control, we don't know what's driving that pattern.
Sure, I mean, in fact, we can make inferences, but we can definitely say that there is a phylogenetic clustering, so that with insularity,
which is because sedges diversified in wetland habitats. So, okay.
Yeah, in fact, this is also something that we are starting to discuss. What does it mean in terms that fens are more phylogenetic clustering in their assemblage, and they tend towards clustering in relation to insularity.
But you don't know that that's due to insularity versus the habitat. Because look, I mean, you've got 3 points. That entire diagram is driven by the 3 points on the left. I mean, do you really, what's the robustness of that analysis?
In fact, well,
based on 3 points?
In fact, here we can see that also the estimate is.
But if you were to do a robustness, if you were to do a randomization test, how much of that is being driven by the leverage of them? So just 3 observations.
But this is randomized. This is already randomized. I didn't say that these are like 999.
So tell me about the 3 points in the far left that are driving that entire blue analysis. What's different about those 3 points? And why are there only 3 points in the far left?
Because we have those 3 which are sites which are less.
So what is being randomized? I guess I don't understand what's being randomized.
I mean, randomized the phylogenetic diversity.
Yeah. Well, I'm talking about the insularity part.
Ah, okay.
So I guess what I'm saying is that this pattern that you're describing here is driven by 3 points out of how many?
No, I agree. But in fact, I fully agree because the pattern is not very strong. But still, we can say that these ones are clustered. These ones, even though the pattern can be.
Which could be due to the fact that you're dealing with fins. open wetlands that may have nothing to do with insularity. That's my point. Is that those
Yeah,
that line that you have there is driven by 3 observations out of however many.
Let me continue with my questions because I didn't finish it once you stole it.
Sorry.
But yeah, I think I can continue with this one. I think more interesting than just the line is that some sites are phylogenetically clustered and some are not, and there's a huge variation. So the next step is to find out if there are any characteristics why a bunch of sites are highly phylogenetically clustered and the other are not, and because we are in the same system, so it's not too much about asking why fens, if the fens are phylogenetically clustered because of insularity. We totally agree that we are probably not able to answer this because you are within the systems and which is already, the flora is phylogenetically clustered, so that's it. But try to explain just the binary of the system. All points above and all belong the line, the dashed lines, if there are any differences.
In fact, this is still this part is all in the random real, and then should be in the 0.95, that should be over dispersion, which is nothing. We have nothing in over dispersion. Because these are percentile, so as I said, I didn't explain too much the methods. So this is kind of, it's not standardized effect size, but it's coming from a randomization of the diversity metric.
So then I'm confused. I'm just asking if we are able to explain why there is such a huge difference in phylogenetic diversity. Why there are some sites which are phylogenetically highly diverse and the other.
No, in fact, I don't know. That's something that we would like to open up, because this is something that it's quite, I think, interesting. And for example, we are discussing also with Ika and Adam, like, I don't know, like knowing that no matter about this, right, but about the fence specialist assemblage are actually clustered. Yes, of course, it's because of the strong filter and the habitat desegregation, of course. But the fact that the outcrops, on the other hand, there is no structuring whatsoever. Can we infer something about the age of the, for example, like of outcrops that we are considering often like as very, let's say, young in comparison with fence, because we have no data since for the outcrops, for example. We don't know if there are these stepping remnants for, I don't know, many thousands of years or even more. I mean, it's something that can open up, like, can question the idea that we are considering outcrops maybe younger than what they are. I don't know if I'm able to, because it's quite challenging, right? Because we are assuming now, also from the previous analysis, that the fens are, of course, the oldest and the most insular.
Maybe we are stuck in one thing, and the presentation was about functional diversity. So maybe this was not the main take-home message. So can we move to another point of the presentation?
No, but for me, I'm happy, because I mean, we need. No, I mean, it's fine. We can keep going also later on. But definitely, it's something that it's puzzling.
So we don't know what's the species pool for the randomization. So I think this we don't.
Just a tiny idea that might answer the Jeff concerns is that, for example, to calculate it based on some fen samples from area like Scandinavia, the habitat is more continuous. So that would, just answer our question if this is an insularity or it just happened conscious.
Okay.
And we have those data, so.
So like another way to interpret this is it could be that this is the dots above the line may be poor fins. And maybe the ones below the line are rich fins. I mean, we don't know.
Maybe if we had the labels of which side that was for you would have been much easier, because for me there are dots and dots there, but probably it's helping. You can ask to explain these
They're all
groups. Sure.
So heat control for that. Okay.
Okay. So another question?
if you deleted this implicitum variegatum, also this pattern disappeared? Or not?
I don't know. I think he didn't check for this.
So if you will
Because it should be somewhere here, right?
That there will be those crops.
Because it should be equisetum variegatum should be somewhere here.
Could be because these are large cross fans all one, like Raksha,
Yeah, I think it's this one. Are those ones? Yes. Yes, I think are those ones. Yes. They go. These three ones, which were here, with equisetum, without equisetum.
Yeah, with the equisetum, I think the only message that I got from that is that it's basically very unstable. So we don't.
But I think I'm not saying that it's good to
too
have
much
both.
trust to the pattern if just one species makes the pattern. It means it's very unstable. So it's not.
Adam was also inferring, he told me that we need to also be aware once we're doing this full phylogenetic control, because it can return some not necessarily meaningful results. So the midpoint, it's a bit more, let's say, conservative.
Are the students who want to ask? Another one? We even don't know which index forces for the calculation. So if there will be some which is using the tops of the
No, actually, that was something that I didn't report. But this is like the index that I showed before is the faith. But then we calculated other three metrics, MNND, MNPD, so like also the relativeness, mean pairwise, and the signal is actually, so either it's more like basal or terminal, the signal was constant.
But the faith is depends on the species reach. So, yeah.
Something different. Something different about serpentines. So if you plan to include also Czech serpentines, so there are also some in Bohemia, so we are sitting on them recently. So I think I didn't notice that you.
Well, no, I mean that was not. They are not only those people that I would like to keep collaborating, but they were like samples because the idea would be to examine, to really measure traits, the whole list of traits affecting the persistence, not only through chronal ability or age or this, but also like the whole thing, like acquisition, everything, pretty much the whole syndrome of this serpentine island syndrome. So I would like to collect at least one trait for each of the major plant functions and test whether this is kind of not only the trait value but also the trait relationship, if they remain constant along insularity gradient, so edaphic island, serpentine on mainlands, on islands, more or less insular, so along insularity gradient, within a biome, let's say Mediterranean, and within a region. And then within the same region, within the same biome across region with California, and then comparing with biomes, with other biomes. So if we assume that there is syndrome that is very much driven by the insularity associated with this super strong edaphic filter, this should be somehow emerging, no matter what the climate. I mean, of course, there should be some variability, but definitely the syndrome should be there, because otherwise, what syndrome? So that would be something that's my idea, like that I would like to develop. That being said, of course, we can examine also not only Tuscan archipelago, but definitely this was at this point in the line what I was thinking how to make the experiment, it's experimental observational settings, how to test some specific hypothesis. But definitely, of course, could be doable. I mean, that could be another biome and another region.
That's kind of mine's more of an easy general question, because we're looking at a lake that has an island. Just talking about what we're looking at, an island in an inland lake, but it's not an island. So it's an island within an island. I was thinking about it when you showed Australia and you had your outcroppings, but you're on an island, obviously, continent. So I was interested in the last part. I was waiting till you showed that last slide. That I would be very interested. It sounds like that's what you're going to be looking at. Is those? Because we're seeing some real interesting stuff. I mean, all organismal, ecologically dispersal within, you can imagine, it's in the middle of Lake Michigan. So there's an island within a lake, which is on an island, okay, but surrounded by a large body of water. It's not salt water, it's great lakes, but all those variabilities, and it would be very interesting to see if you did some comparisons there with what's going on in Australia, Outcrops, here in the Czech Republic.
The thing is that in Australia, during my PhD, I was more into conservation part, so using traits for identifying refugia. So instead, and I didn't use at all like biogeographic predictors or approaches, notions. But still, I was working with islands, with interact systems, and then here I tried to combine the things. But definitely, this looks like nice. I don't know, but how many islands do you have in such?
In that type of condition, it's kind of unusual in the Great Lakes region, but they do exist. It started off as a fisheries, believe it or not, looking at fish, and there's probably, I would say, 5 or 6 within that region like that. So they're not a.
So that you can have like a bit of replication.
And then what's inland, like you described, how far the distance from your outcrops from the water, the ocean, or dispersal mechanisms. We're looking at the same thing in those island systems. So it'll be interesting. I haven't seen any of the results.
Yeah, because this idea about the serpent is in fact to have island on island.
Exactly. So when you showed that lab, I was like, cool. So for future work, interesting. So just a comment on it.
Okay, your last round.
Thank you for your presentation. When you were talking about in the last part of that functional diversity of the low ground traits decreased with insularity for colonial species, this decrease was always biased towards some kind of particular trait, or it was just random?
The diversity, as I was saying, is how many organs pretty much plant species can have. So it's calculated that way. So it's not telling us we can eventually go and look at, which one was the organ that was striking. But otherwise, it's like, especially because type of prone organ is categorical, so then we measure we use covert distance for the metric. So we don't really know. From that analysis, which one is consistent. It's simply that there is less possibility to have more diversified organs or more organs in this case.
Thank you. Thank you very much for your presentation, for this discussion.
Thanks for
all of you for